today we're continuing with cellular respiration, and we'll be looking at the first main step, which is glycolysis. Okay? So this is going to be the first step or stage to cellular respiration. So what is glycolysis? Well, if we look at the word glycolysis, lysis, hopefully you remember, it means the breakdown. So we're actually going to break down sugar or break down carbohydrates in this process. So what we're first going to look at are the main reasons, the who, what, when, where of what happens during glycolysis. Because remember, this is just the first step. So we're not yet creating lots of energy. We've not yet reached the end of cellular respiration. But we have created some, and we're starting the process to create more ATP, more energy. So when you look at the who, this is actually going to happen in all organisms and all cells, because this will also happen in prokaryotic cells. Because it happens in all organisms, this is known as a conserved process. If you think back to our cell unit, about the unity of organisms, this process scientists have recognized as something that happened with the very, very first cells. So all cells have done this process. All cells have done or have performed glycolysis as a way to break down carbs and begin that process of converting it into energy. So where does this happen? Well, if you think about our cell types, not all cells have mitochondria, which is where we usually learn that cellular respiration occurs. But because not all of them have mitochondria, it can't happen there. This process can't happen there for all of them. So this will actually happen in the cytoplasm. This first stage of cellular respiration actually happens in the cytoplasm of the cells and not the actual mitochondria itself. So what happens here? Well, we said that we're breaking down carbs, we're breaking down sugar. So we'll have actually what's known as a 6-carbon sugar. And this will be broken down into 2 3-carbon sugars. 3-carbon sugars. And I'm writing breakdown in the middle. Okay? You have 1 sugar molecule, 1 glucose molecule, and you're going to break it down basically in half into 2 3-carbon sugars. Now, why does this need to occur? This process has to occur because we are actually trying to transfer electrons to a carrier. Okay, we're trying to transfer electrons to a carrier. So this goes back to the idea of redox reactions. We have NAD+, and we're actually going to add to it 2 electrons and a hydrogen, and we'll produce NADH. When we do that, what this means is that we're actually oxidizing glucose. Glucose is going to be losing those electrons, and we're actually reducing the carrier. Reduce carrier, which is NAD+. Okay? Remember that the whole idea of this, the main reason why we're doing this, is to eventually produce as much ATP as possible. When you're looking at the ATP molecule, remember that the energy is going to be found between the bonds of those phosphate groups, of those 3 phosphate groups. So this is where the energy is found. What's going to happen is we're actually going to start with ADP and then some diphosphate, and we'll add in that 3rd phosphate group. then our cells can use that as energy when we break that bond. So glycolysis happens in actually many, many steps. And remember, again, this is just the first step to cellular respiration. This isn't the complete package. Not everything has happened yet. So glycolysis itself happens in about 10 basic steps. We don't expect you to memorize all 10 of these steps, but there are 3 major portions to this process that you'll have to be able to recognize and understand. So, first things first, let's talk about our start point and our end point. Start, and here's our end. When we start this process, we're going to start with glucose. And glucose is going to be our 1,6-carbon molecule. 1,6-carbon molecule. Okay? That's what we start with. At the very end, we're going to end with a molecule known as pyruvate. And pyruvate is actually our 3-carbon molecule. Pyruvate itself is composed of just 3 carbons in its backbone, and we're actually going to make 2 of these molecules at the very end. So 1 glucose at the beginning, 2 pyruvates at the very end. Now, when we're looking at this process itself, we actually first want to look at these first 3 steps. So the first 3 steps are separated right across the top. And what you actually have occurring in these first 3 steps is actually known as an energy investment period. So what we're doing is we're actually going to be using 2 ATP molecules to change the shape of glucose to secure it in the cell. So glucose itself, if you'll remember, is a fairly big molecule and can't diffuse easily through the cell membrane. So we actually have transport proteins that will carry this molecule through the cell membrane. But we also don't want it to leave the cell membrane once it has entered. So instead, what we'll do is we'll actually use 2 ATP molecules. To change the shape of glucose into a form that cannot be transferred by those transfer proteins. And so we're going to use or invest energy to do that. So what we want to look at first are, well, we actually want to look at 2 enzymes here. You have this first enzyme called phosphofructokinase. And this is what's going to actually be known as a commitment step. The reason why this is called a commitment step is because we are using energy, ATP molecule, in this process. And if we actually don't end up needing to make more ATP, then we're not going to use or invest the energy into that process to make more ATP. And we'll talk about that form of regulation in a little bit. The other thing that we want to look at is isomerase, another enzyme. Remember, ACE hints to us that it's an enzyme. An isomerase, an isomer, if you'll remember, has the same formula, but a different shape. And you'll want to write that in a little neater than I did. So, same formula, still 6 carbons, but we're actually going to change the shape. So that should make sense because we're changing the shape to secure it in the cell. Okay, we're talking about that glucose molecule. Then what will happen is after we make this molecule, this new molecule, this different shaped glucose molecule, you'll actually see two things happen, or two products being formed. You have a dihydroxyacetone phosphate molecule. This molecule is a 3-carbon molecule, and you'll actually see that it has to go through this intermediate step, another isomerase process, isomerase reaction, to form glyceraldehyde 3-phosphate group. And so this is actually what both of them will become. This is another 3-carbon molecule. Notice that first we make two separate molecules, two different shapes. This one has to go through step 5 to eventually form this same molecule. So eventually you end up with 2 of these. Okay? So you end up forming 2, and what we actually call this G3Ps, glyceraldehyde 3-phosphate, 2 G3s. These will actually eventually get oxidized during our reaction, during the rest of our reaction. And then we will create 1 NADH per every G3P. Meaning with each of these G3Ps, we actually get out of it 1 NADH. Since we make eventually 2 G3Ps, we'll end up making 2 NADHs. So at the end, we'll have 2 NADH molecules. So then as this process continues, you want to notice that here we have ATP actually being formed. Going through this process some more, going through some more enzymes in the cytoplasm, you'll form another or a second ATP molecule. So at the very end of this process, we actually have 2 ATP molecules per each G3P molecule that's formed up here. So eventually that means that we'll have 4 ATPs total that are being made. But remember, we did use 2 ATPs at the beginning. So in the next part, what we want to look at is the main idea of glycolysis, a little overview of the process itself. So the main idea of glycolysis, first of all, we want to remember that glycolysis is going to happen in all cells. It's going to happen in the cytoplasm or cytosol of these cells. And at the very end, well, we start with, 6-carbon molecule, and we'll actually end with 2 3-carbon molecules. But we're also producing these 2 NADH molecules and these 2 ATP molecules net. Okay? So your net gain, because you used 2, but you made 4, we used 2 ATP molecules, we made 4 at the end, so our net gain is 2 ATP molecules. And then in this next process over here, we again have this image of what is our net molecule. Here we're using 2, here we're gaining 4, here we're gaining 2 NADHs, and at the very end, here's our net gain. We also notice have some water that's being produced, and that will be used later, but we're still going to concentrate just on the pyruvate, the carbons, the ATP, and the NADH. And then we'll actually talk about how this gets regulated later on in class.